Oh, yeah. You missed the world ball.
All right. So, Gray, do you want to give these out? Wait. All right. Who wants to claim this?
Hope it's a 20%. I was like,
No, 28 over 30. 28.5 or 30. Who wants to claim it? No one? So take it back. Seems like nobody wants to claim it. You get 0.5 points to 29 if you claim it. And it's correct.
oh.
And if it's correct.
Then we get minus.
What's that?
I don't even know.
Last going once. Going twice, huh? Going last.
Is that person here?
Yes.
Okay, everybody raise your hands. Ready? Yeah. Go. I don't think it. I don't. That's past enough. Yeah.
Nobody wants that half a point. Okay.
Oh.
Love the half.
Wow.
What?
No way. Have to come back.
Love the excitement. I don't know. Why did he say that?
So mean.
Minus 5 points.
He asked us, how do you think you did on the quiz? And I said, I feel like I did well. And then
And he wasn't expecting that, he said. All right. Of course, now you're distracted. You're looking at the quiz. I'll give you a moment, huh?
we're fine.
Mm-hmm. Doesn't want to see it. Yeah, almost a full house. William is missing. Right? Nobody else is missing. All right, while Gray's doing that, make sure you're in the lab by 9:30 sharp on Wednesday. We'll do 50 minutes lecture and then review so that we only do maybe 10-15 minutes in class lecture and then we party.
On to that.
Lab. Yeah, I don't know if this room has people in. So, yeah. Okay. So, just a quick recap. I'm not going to go over all these questions, but make sure as you study, you review these questions as a study guide for minerals determination. But very quickly, why is it important to analyze mineral?
Yeah.
Nutrition label. Anything else?
Yeah, 6.
Yeah, tell me. One more thing. Health claims, absolutely. If we're doing fortification or enrichment, so we need to do that. What else? So minerals impact processing. So addition of calcium to milk for cheese making, determining hardness of water because it will impact processing parameters. So all of that. So review these things as you study. Okay. Just another quick one. Name mineral analysis methods, or one or two. Titration. Huh? Atomic absorption or inductively coupled plasma atomic emission spectroscopy, and then colorimetric. We'll talk about that in a little bit. So titration, colorimetry, absorption or emission. Okay, very good. Then you review the others on your own. So colorimetric methods, this is usually done for specific minerals and only if we're analyzing one type of mineral, and it's very common for phosphorus. So this is done where your phosphorus react with chromogen like molybdenate, and it gives you a very nice yellow color. So obviously you will run a standard curve and you would follow Beer's law. And it's very common for phosphorus, more common than other. There is another colorimetric method for copper, but yeah, only if you're looking at one particular mineral that can react with to form a chromogen with a reagent. The last one method is the ion selective electrode. So it's similar to pH meter, but the electrode, instead of it being sensitive to hydrogen ions or hydronium ions, it would be sensitive to specific minerals or elements. Common ones are calcium electrode, sodium, chloride, potassium, and fluoride. So again, it's the same principle, but that electrode will be sensitive to these particular elements. Some considerations. Why is it not working? Okay. Some consideration. Sometimes there could be a cross-reactivity where the electrode can be responsive to other ions, which will cause about 10% error. You would also need to run kind of standards alongside to determine the concentration. It's very sensitive, but sometimes when you have really low concentrations of element, you need to put the electrode for a long while to get equilibrium. Yeah, exactly, but the electrode is specific for the element instead of specific to hydronium ion or H plus, it was specific to calcium or sodium.
But is it like a separate machine or?
No, it's the same pH meter, yeah, but then you change the electrode. Yeah.
And the
Probably-
measurement system, or
Huh?
the measurement is just a number.
Yeah, it will give you a concentration, but then you will run a standard. Yeah. You will relate the measurement to a concentration. Yeah. Okay. So it's commonly used in processed meat, determination of calcium in milk, salt in butter and cheese, potassium and sodium in wine. So these are the common application for these electrodes. Now I want us to solve this together. I want you to make an effort in understanding this problem. It may or may not show up in the final.
What's the time on it today, or
Huh?
are we like turning this? either?
I'm gonna work with you on it, so you don't have to turn it in. You turned in something last week, right? Yeah, that's fine. But I want you to learn the concept really well, take good notes, ask questions as we solve it together, because I want to move on to the next lecture. Okay. So in this question, we're saying that we're determining salt concentration using the backward titration. Oh, let me remove that floating thing, annoying thing. All right. So we're doing the backward titration, which is the Vollard titration, determines salt. And as a reminder, I'm putting up here the chemical reaction. So we put excess silver nitrite to react with whatever, how much we have salt, mostly Cl minus. So we'll get silver chloride. And then the remaining silver nitrite will be titrated with thiocyanate. to an endpoint that is red. Okay, so basically when you have excess, you would put a known volume of that silver nitrate. And then whatever is remaining, you will titrate it. And then the difference of that silver from the original to what was left in excess will be the amount of moles that reacted with chloride. So, repeat. Yeah. So when you put a known number of moles, a known amount of silver nitrate, part of it will react with the chloride. And then you will have a remaining silver nitrate that did not react. So you would titrate that. And you would know how many moles that was remaining. The difference in moles from the original and what is left would be the number of moles that reacted with the salt. And since it's 1-to-1, so number of moles will be equal to number of moles. So we're solving backward, right? So here I'm telling you that we needed 30 milliliter of 1.1 molar silver nitride. And then the residue was titrated with 3 ml of 0.1 molar potassium thiocyanate. I want to determine the number of moles I had originally, all the silver nitrate that I added, and then I want to know the residual that remained, how much of it remained, so I would need to calculate number of moles. And then get the difference. The difference would be the number of moles that reacted with equal number of moles of chloride. So that's the back calculus. All right. Did I lose any of you anywhere? You can ask, it's fine. I can repeat. Oh, golden is missing. Oh. I knew that there was one more missing. Okay, so question. So we get the total number of moles, 30 ml of 0.1 molar. I need to get the total number of moles in 30 ml. And then it's 1-to-1 mole also in silver to thiocyanate. I needed 3 milliliter of 0.1 to kind of titrate the excess, the remaining. So I want to calculate number of moles here. The difference will be the number of moles that reacted with the chloride. And since it is the same 1 mole to 1 mole in the chemical reaction, then the number of moles reacted will be equal to the number of moles of chloride. Okay, so then first I said 25 grams of food sample was dried, then ashed, and finally analyzed for salt. The weight of the sample, the weight of the dried sample was 5 grams, and the ash weight was 1 gram. Okay. So what was the moisture content? Let's solve that. I'm sure. Okay. Fill out a piece of paper for us, please. All right. Good. I need to do it.
80% moisture content.
Huh?
80%.
80% moisture content. Thank you. That was very quick, but yes. Make sure you put in the calculation to get 80% moisture content. All right, work on the ash on dry basis then. All right, what's the ash on dry basis? 20. Very good, very good. All right. So moisture content is 80% because 25 minus 5 is 20 grams of water. Okay, that you evaporate it. 20 over 25 times 100 will give you the 80%. And then ash on dry basis, that would be 1 gram over the 5 grams of sample. Because 5 grams is dry matter. That would times 100 will give you the 20%. Are we all good with that part?
You just want the one scale.
Yeah, do everything. Yeah.
I'll do everything.
Yeah, A, B, C. Yeah. All right, so the moisture show calculations and ash show calculation. Now you need to determine salt. All right. How can I get the mole, the number of moles of 30 ml of 0.1 molar silver metal? Yes, you're right. So we have 0.1 molar, which means 0.1 moles per liter, which is how many milliliters? 1,000. But now, what would it, how many moles I would have in 30 ml? Point 1 moles over 1,000 times 30. Point. So in 30 ml, I have, so we have altogether, we have 0.003, correct? Moles of silver. That's my excess. That's it, all of the silver. And then I'm telling you that, okay, I needed 3 milliliter of 0.1 molar potassium thiocyanate to reach the endpoint. And again, it's 1-to-1 ratio, the chemical reaction. So how many moles are there in 3 ml? So this is the excess, the remaining, sorry, the remaining, not the excess. This is the excess. And then this is the remaining after silver reacted with chloride. So if I took 3 ml, it would be 3 ml times 0.1 moles in 1,000.
0.0003.
Yeah, so 3 zeros. Now what do? I have the remaining, I have the excess. Now what do? Subtract. So 0.003 minus 0.0003 will give me 0.0027 moles that reacted with chloride. Are we all following? Yes, Emma?
First was the excess.
What's that?
The
The
first-
excess, yeah. So what I took my ash, I added 30 milliliter of 0.1 molar. So that means I added 0.003 moles of silver. And then after the reaction, I titrated with potassium thiocyanate. And I needed only 0.0003 moles. So the difference would have been the moles of silver that reacted with the present chloride. And since it's 1-to-1 ratio, so this is the moles of sodium chloride that I have. So moles of silver, you all see this? Moles of silver equal moles of NaCl, because it's 1-to-1 ratio in the reaction. So if I have 0.0027 moles that reacted, this means I have 0.0027 moles of NaCl. Can you convert that to grams? How would I convert 0.0027 moles of NaCl to grams.
It's molecular.
Yes, absolutely. So if I want to get the mass of NaCl, it would be number of moles times the molecular weight. So number of moles is 0.0027. What's the molecular weight of NaCl? In the exam, I will give it to you.
54.
Yes, so 35.5. No, wait. Yeah, 35.5 plus 23 will be 58.5. Yes. I'll give that to you. You don't have to remember. So grams per mole. And the unit here is mole. So 0.0027 moles times 58 gram, 58.5 grams per mole. So moles cancel. So what would be the weight?
0.157.
0.157.
768.
So I will round to 0.158 grams. Now, what was the question? It's asking salt content in the original sample, expressed weight by weight. The original sample is before drying.
158 divided by 25.
Times 100. Percent. Weight by weight, percent. Yes. So what would it be? You got it. Okay. Okay. See, it would be 0.158 grams by 25 times 100, give you 0.632%. Okay. You all followed this? Okay. Anything you want me to repeat? Good. All right. We close this chapter and go to another very good and exciting chapter. I feel like you never believe me anymore. Okay, it's for every chapter except. No, I do like carbohydrate. What's wrong? Why? I only joke around with George. There's only really one chapter that I really dislike. You guess? Can you guess? No, it's fun. That one is fun.
Protein.
No. Oh, no. It's just happened 2 years in a row. I was gone for the vitamin lecture. It's just that. Come on, guess.
Sample top.
Oh, okay, then there are two of them. Yeah, I really dislike that chapter. It's such a boring chapter, but very important, unfortunately. Where did I put my clicker?
Oh, it's over here.
Thank you. Do you know that one time I put my phone in the fridge and my daughter found it? Mom. Okay. Joe, you didn't guess? Okay. Oh no, it's pretty cool, huh? No, it is fine. It's okay. Fluorescence. Yeah. It was for me, physics was never a thing for me.
Same.
Yeah. Yeah, but I learned it over the years, but I still don't like it very much. Okay. Okay. Why does this keep showing up? I feel like all right, so Immunoassays, very, very useful methodology in food analysis. We use it in so many different ways that we're going to give a lot of examples. But when you hear immunoassay, what does it mean to you? What is the immunoassay about? Right, it's called immunoassay. Immune system. Warmer? Getting warmer? What's in the immune system we use in analysis?
White blood cell.
Oh, no, we don't get white. Antibodies. Wonderful, wonderful. So antibodies react with something in these assays. What is it called usually? When antibody, we mean what? Has some letters. Well, it does, it is used for bacteria and viruses, so you're not very wrong there. But what is usually the component name that reacts with the antibody in an immunoassay? Have you heard of the term antigen? So it's immunoassay is a reaction between an antigen and an antibody in a way that will be able to either get a qualitative observation, change in color on a gel, or we get quantitative data using a very common assay known as, Say that.
No.
No? If I say it, you're gonna go, ah. Have you heard of ELISA?
Oh, I remember that.
Do you know what it stands for? E for? No. Well, there is immune. There is an immune in there. E?
Enzyme.
Enzyme like immunosorbent assay.
How did you know that?
Oh, I'm just awesome.
Okay. So ELISA allows us to do quantitative and sometimes semi-quantitative, depending if we have standards that we can quantitate. And there are other immunoassays like Western blot. Have you heard of Western blot?
Yeah. Yes.
Have you, okay, we're going to talk about Western blot, but also have you, I give this example a lot, but I'll save it for later. Okay, I'll save it for later. Anyway, so immunoassays allow us in food analysis to determine antigens. The antigens can be what type of antigens, for example? What do we analyze with ELISA? Proteins that are allergens, for example. So allergenic proteins, GMO proteins, authentication of protein sources. For example, have you heard of this case a few years ago where I think somewhere in Europe, maybe England, they had horse meat in the burger? Have you heard of it? So you can do immunoassays such as Western blot to detect whether or not you have type of protein that should not be in the beef, along with the beef protein. And it's also used for detection of small molecules like antibiotics, for example, or aflatoxin. It's not necessarily proteins. You can use them for small molecular weight toxins and antibiotics. Also can be used for detection of bacteria and viruses that will have the antibody reacts with the protein on the surface of those bacteria or viruses. It's very, very useful assay in food analysis. Yeah. So I don't know why this is not wanting to work. Okay. All right. So the concept is you have this antibody and then you have antigen. So the antibody is really a protein, believe it or not. So antibodies are made of 4 protein polymers. So you have 2 long polymers and 2 small polymers. And this area over here at the top is where the antigen binds to the antibody. Okay, so antibodies, for example, they are produced. Okay, sorry about that. Okay, so antibodies are produced in our body within a specialized cell, type of cell called B cells. So you know how people get allergic reactions? It starts with the individual would get sensitized to an antigen. So let's say we have a peanut allergy. So let's say an infant has a peanut allergy, and the first time they have peanut, they won't react, but their body will produce antibodies because they assume that this peanut protein is a foreign body. The next time this baby gets peanut will flare up because now in their system they have antibodies that will react with this foreign body and will cause this reaction. All right, so these B cells, and then the antibody has a very strong interaction with the antigens. All right, so this is a structure of an antibody, immunoglobulin G, one form of antibodies. And the affinity between the antigen and the antibody is really very, very strong, but non-covalent interaction. However, it's a really strong interaction between ionic, hydrogen bond, hydrophobic, van der Waals kind of interaction, but not covalent, but still very, very strong with a very high affinity constant. All right. Have you heard of the term epitope? Okay, so remember it. An epitope is the region on the antigen that the antibody is specific to. So it's where the antibody binds to the antigen. Is called the epitope. Now, if we have a protein molecule, erase this. If we have a protein molecule, it's a really relatively large antigen. So a relatively large antigen will have multiple epitopes. Okay, let me just illustrate. So if you have protein, you have a relatively really large complex protein. And then you can have multiple regions on that protein where antibodies can bind. So your B cells in the body, once they are exposed to a protein that they consider is a foreign body, they produce different types of antibodies that can react to different region on the protein. Okay, so epitope 1, 2, 3, 4, could be more. Okay, but we have different types of epitope, linear epitope and conformational epitope. What does that mean? It means that linear, you have amino acid in a row, in a sequence, 1, 2, 3, 4. These together form an epitope. So these 5 amino acids react with that binding site. Sometimes your epitope is conformational. So a region of the protein, not in sequence, with another region just away from it, but it just, the shape made them close to each other. They're not necessarily in sequence. The amino acids are not necessarily in sequence. They're just how the protein folded made them close to each other, and then they became a region where the antibody can bind. So this is called conformational epitope. Then another thing that I wanted to explain to help you understand the assays later on is polyclonal versus monoclonal antibody. And what does that mean? So this particular example, when an antigen is introduced to the body, the B cells, you have many different B cells, will produce antibodies that could react at different sides of the antigen. Sorry, it's my sensor. Okay. So these are polyclonal, meaning they're not the same. They don't bind the same epitope. They bind different epitopes. So usually the body produces polyclonal because you have many different B cells, differentiated B cell. So you will have polyclonal. So, but if you want to buy ELISA reagents standardized and you want to carry out a kit, an ELISA kit, usually the ELISA kit has a monoclonal antibodies, which would be better for assays to have monoclonal antibodies. What does that mean? That means all of the antibodies will react to a singular. So you will have antibodies that are exactly the same, and they will only react with this region, monoclonal. So, and I'll tell you where it's important when I give you examples on different assays. How do we produce monoclonal? Not in a body, not in somebody's animal body or get sera from animal or human. This is produced by cell culture. You get one type of B cell and you fuse it with a cancer cell so that it proliferates faster. And it will produce one type of antibody only, because it's one type of B cell that will produce only one type of antibody. This is very efficient for ELISA kits. Whereas if you want to get polyclonal, usually we do this for allergenicity studies. So if we want to determine how allergic an individual would be for a protein or a modified protein, we want to make sure that we get sera from that allergic individual that contains polyclonal antibodies that would react with all possible epitopes. So if we're doing allergenicity study with a human subject, we really need serum from allergic individuals that has polyclonal antibodies. But if we're doing assays to detect presence of a particular antigen, usually it's best to get the kit that has a monoclonal antibody. And I will give examples later, but I just wanted you now to know the difference between polyclonal and monoclonal. Again, poly means you have antigens or antibodies, that will react at different epitopes, whereas monoclonal, they are the same antibody that will react with the same epitope. All right, very good. So immunoassays, like I said, are great because they're very, very specific, very sensitive, and very simple. We just have to find out the appropriate antibody for the antigen that we want to measure, whether the antigen is a protein or the antigen is a mycotoxin or is an antibiotic. So the other consideration is we need to differentiate between a free and a bound antigen, because when we have a reaction, we have our antibodies, and then we have excess antigen that will react, and some antigens will not react because they're excess. So we need to have an assay that differentiates the bound antigen from the non-bound antigen, and I will explain that more later. Another important feature of proteins, and antibodies are proteins, is that they bind to hydrophobic surfaces. And that is important because in ELISA assay, we usually run it in a plate so that has 96 wells. Have you seen a plate with 96? Yeah. So this kind of plasticky thing that will have high surface hydrophobicity, and the protein binds really well to that. And this is an important consideration. So if we want to get usually polyclonal antibodies, we either get them from a human if we are determining allergenicity for human studies, or if we just want to get polyclonal antibodies to use in our assay, we can get them from an animal. So we can inject the animal with the foreign body and then collect serum after a little while, and the serum will have antibodies for that antigen. So I think we talked about applications. We talked about detection of allergen, meat species authentication, GMO, toxins like aflatoxin or other mycotoxins. Sometimes we can detect specific pesticides as well using ELISA, antibiotics, and then identification of bacteria and viruses. So a lot of different applications for immunoassays. So we talked about ELISA being enzyme-linked immunosorbent assay. A mouthful, but it's good just to remember ELISA. But there are different types of ELISA. There is the sandwich ELISA, which literally is an antigen sandwiched between two antibodies. That's exactly what it means, sandwich ELISA. An antigen sandwiched between two antibodies. Indirect ELISA, I'll explain that in a little bit. And then competitive ELISA. I'll talk about each one of those. And then we have other techniques where we use immunoaffinity for purification, like chromatography, affinity chromatography, lateral flow strip, and that's an important application for quick qualitative determination. If you're in the field and you want to know GMO present or not, you can do lateral flow strip test. Western blot is qualitative, but very useful for screening. Okay. So the enzymes that are usually used in ELISA are very active enzymes, so that you have a quickly action within half an hour. You have a really good high reactivity, and these are usually either horseradish peroxidase or alkaline phosphatase. These are usually the enzymes that we link to the antibody or the antigen. Why do we need those enzymes? Because usually once those enzymes are linked to the antibody or the antigen, we add a substrate. And the substrate will react with this enzyme, and then we get a product that is a chromogen. So we can do absorption studies. So it's basically the role of the enzyme is to convert the substrate to a product that gives us color. The more bound enzymes are there, the more color we get. So we need to use stable, highly active enzymes that we can link very easily to an antibody or an antigen, and it readily catalyzes the conversion of a substrate to a color compound. And then often we use a plate reader because you would want to run standards, you would want to run multiple replications, you want to run multiple samples, and then you can do all of that in one single plate that has 96 wells. And then there is a plate reader like a spectrophotometer. Instead of putting a tube, you put your entire plate, it goes in, and you get a reading for every well. Very, very effective. All right. So a few terms that I want you to know about enzyme-linked immunoassay. So we have direct detection and indirect detection. So direct detection is when the antigen react with a labeled antibody. So it's direct. So the antibody that is specific for this antigen is labeled with the enzyme. So if it's labeled with the enzyme, then you can add a substrate and then it changes the color. So that's direct. Now the indirect, and then you would go Y. I'll tell you in a minute Y, and then we'll end. So the indirect is you have your specific antibody to the antigen, but it's not labeled. What is labeled is an antibody to the antibody. Why do we even do that? So an antibody to the antibody, let's say this is a human antibody. We inject an animal with the human antibody, the animal will produce an antibody to the human antibody. No, the antibody becomes an antigen. This one gets labeled. Now you would say, why? Why don't we label directly? Because it's much more work in the lab to get those antibodies that you're looking for and to label them yourself. You'd rather get a reagent that is already labeled that you can purchase. So then this makes your life easier in the lab where you have your antibody, you have your antigen, and you just purchase a labeled antibody that can react with the antibody in your study. And that labeled is the enzyme, and then you get enzyme reacting with a substrate, and then you get a reaction, you change color. So that's the only difference between direct and indirect, and that's because you purchase an already labeled antibody. So you have two antibodies in that. And the primary antibody would be the one that reacts with the antigen, and the secondary antibody is the antibody that reacts with the primary antibody, and this is the one that is labeled. Okay, so armed with this knowledge, on Wednesday, we'll finish the lecture in lab, 9:30, don't forget. And send your baby pictures to Emma. Okay. Thank you. Please, when you come on Wednesday, if you don't mind, review some of those concepts so that we can go fast.
All of the I can review other sheets. I have not done a single one yet, and I
Can you go over the indirect
this one.
Yeah,
again?
yeah.